सब्सक्राइब करें हमारा YouTube चैनल, बेल आइकन को क्लिक करना ना भूलें, जिससे आगे आने वाले सभी YouTube अपडेट्स, वीडियोस, नोटिफिकेशंस आपको मिलते रहें।
Hello everyone, welcome to the Parishkar family. This is me, Om Prakash Bishnoi. Today we will discuss the model test paper for biology. Okay, in that model test paper, let us discuss first some instruction that is provided by the CBSE. So all the questions are compulsory. In that series, there are 5 sections, and in these 5 sections there are 33 questions, and these 33 questions are divided into 5 sections. So first section contain the 16 questions, second section that is the B section, and that contain the 5 questions, and that third section which contain the 7 questions. The fourth section contain there are 2 case based questions, and the last section which contain the 3 questions. Okay, so that is the clear cut demarcation of that distribution of questions in different sections. Okay, so they are divided into different parts. So let us discuss the first section. What is that first section is about? And this first section is of multiple choice questions. So there is no worrying thing about this. There are 4 options are given, and out of these 4 options you have to select one. Okay, so first question is, which of the following statement are correct with respect to hormones secreted by placenta? So look at this one, that is the hormones which are secreted by the placenta. Now look at the first option. Placenta secrete relaxin hormone, and that relaxin hormone is in the later stage of pregnancy. So what is the role of that? We will see it. Second option is placenta secrete high amount of follicle stimulating hormone during pregnancy. Third one is placenta secrete relaxin during initial, look at this one, this one is the relaxin initial stage of pregnancy. And last one is placenta secrete HCG or HPL during the pregnancy. So these are the options. So look at this one, which one is the correct answer? Now the correct answer is that the placenta secrete relaxin during the later stage of pregnancy, and the fourth one, placenta secretes the HCG, and so option 1 is correct one. Why? Because look at this one, relaxin hormone, that hormone is very much playing important role, and that role is in the parturition. Second one is HCG, that hormone is secreted by the placenta to maintain the pregnancy. Okay, so because of that reason, the option first and the fourth one is very much correct one. Now second question is, a female undergoing IVF, in vitro fertilization treatment, okay, and that is because of blocked fallopian tube. Now in the reproductive system, fallopian tube is very much important one, and these are those tubes where the fertilization is taking place. The technique by which the embryo with more than, now look at this one, they are in vitro fertilization means that is infertile, and when that women is infertile, that means you have to transfer from outside. So in this one where the blocked fallopian tubes are found, in these one embryo with more than, we are transferring here complete embryo, there is no transfer of cells, and there is no transfer of that gametes, there is transfer of embryo with more than 8 blastomere. That means there is initial development was started outside in the embryo, will be transferred into the female for further development. So which stage we have to transfer? So that is the correct, which one is the correct answer, and that is the answer of IUT, intrauterine transplant. Now third question is, which of the following statement indicates parallelism in genes and chromosome? So that is talking about the genome. So in this one, look at this one, gene and chromosome. So the genetic material in eukaryotic cell or in prokaryotic cell, genetic material is divided in the form of chromosomes. So in prokaryotic cell it is present as a single molecule, while in eukaryotic cell it is present in multiple copies, and that is known as chromosomes. Now those parts which are determining the particular character, they are located on the chromosome, and these sequences are known as genes. So what is that parallelism, how they, because chromosome are transmitting from one generation to next generation, then how these genes are transmitted? So look at this one. Now they occurs in pairs, they segregate during gamete formation, third one is they show linkage, and fourth one is independent pair segregate independently. So look at the imagine about the chromosome. What is the chromosome and what is that particular structure of chromosome, and in that chromosome how these genes are located on that particular chromosome. So if you are able to analyze these things, then you are able to make a connection between the genes and chromosome, and after that when you are able to make the parallelism, that parallelism means how they are transmitted together from one generation to next generation. So look at this one. Now answer is fourth one. So first, second and that fourth option is correct. Now they occurs in pair. Look at this one. If a cell is diploid, that means in diploid cell there is 2n condition, and in that 2n condition there are 2 chromosomes, and there are 2 chromosomes, that means there are 2 genes are located on the 2 chromosomes. So first thing is correct, they occurs in pair. So chromosomes are also present in pairs, and the genes are also present in pairs. Second thing, they segregate during the gamete formation. Now gamete formation took place because of meiosis, and in that meiotic division there is segregation of chromosome, because during gamete formation that diploid cell is converted into the haploid one. So gametes are haploid. So gametes contain one complete set of chromosome. Now in that complete set of chromosome, now these gametes become the haploid one. And when gametes become the haploid one, so there is a segregation of chromosomes. Now if there is a segregation of chromosome, in the same thing there is segregation of genes also. Now fourth one, they show linkage. So chromosomes never show the linkage. Okay, chromosomes are they are segregated into different gametes, but here linkage is shown by the genes, because those genes which are located on the same chromosome, they are trying to transmit from one generation to next generation together. Okay, so this character is not found in the chromosome, that may be found in the gene. Okay, now fourth one is independent pairs segregate independently. Yes, because during the formation of gamete, now look at this one, 2n means there is one set, and that is one set. That one set any organism will get from the father, and that is one set will get from the mother. So when they come together, that means every species have a fixed number of chromosomes. One set is coming from mother, one set is coming from the father. So whenever there is a mixing, at that time, after that mixing, they go further in the gamete formation. At that time, it may be from this set to the genetic material go to this set, from this set to this set. So that they are segregate independent. That is possible. So that is possible also in genes. Okay, so the first, second and the fourth option are correct. So correct option is first, that D answer is correct. Now look at this next one. Evolution is a continuous process. Who proposed abiogenesis first, but biogenesis ever since? Now look at this one. That there is a definite beginning of life, and after that definite beginning the evolution took place, and there is a huge diversity of life forms emerged on this earth's surface. When the huge diversity emerged in this one, what is that mechanism? So if we go to the basic level, and that basic level is cell, if we go to that basic level of cell, then after how that formation of cell took place, and look at the diversity of biomolecules which are present in the cell, and those biomolecules which are converted into form of mechanism. So look at that mechanism complete. So who proposed this mechanism? How that took place? How that these cells are organized? So look at this one. Who proposed this one? Operin, the first one is Operin Halden, B one is Miller, C is Darwin, and Louis Pasteur. So option correct is Operin Halden. So because they said that there is a chemical evolution, and in that chemical life, that after that chemical life is converted in the form of cell, and that cell is later on evolved in the form of diversity of living organism. So now look at this one. Now the fifth question is the coconut water tender coconut is. So in that coconut, what is that liquid part? And that liquid part is, that is the answer is the B, free nuclear endosperm. What is that endosperm? Now in this one, because this endosperm is a nutritious tissue. This endosperm is a nutritious tissue which provide nutrition to the embryo. Okay, and how that is formed? That is the formed because after fertilization, when that fertilization took place between the male gamete and the egg, the second nucleus of the gamete and the second nucleus of the female embryo sac, they fuse together, and they fuse together and form a nucleus of 3 end. That means triploid condition, and that triploid nucleus or the cell will multiply and form a tissue, and that tissue will used by the plant for the development of embryo in the seed. Okay, so that is the endosperm, and coconut is the endosperm. Now there is one very much important question. Psychosis and neurosis are symptoms of following disease. Okay, now in this one, which of the following disease exhibit these two symptoms? So that is by the B1, that is a borderline personality disorder. So that character is shown by this disease. Now seventh one question is the biochemical oxygen demand. So that biochemical BOD, that biochemical oxygen demand, what is that demand in the biodegradation degradation of biotic material in the water conditions? That is the demand, and how that is calculated? That biochemical oxygen demand is calculated. So that option D is correct one. Dissolved oxygen multiplied by the dilution factor, and that is the characterization of the biochemical oxygen demand. Now eighth question is, a biotechnologist wanted to create a colony of E. coli. So in that E. coli there is a plasmid, the plasmid BR322 sensitive to tetracycline. Okay, which one of the following restriction site would he use to ligate a foreign DNA? So in biotechnology we are able to incorporate the outside gene. Because of restriction enzymes, we are able to incorporate that the outside gene into any another genome, or in this one, the plasmid are acting as a vector. So these plasmid are found separated mostly from that bacterial cell, and that is the artificial plasmid that is developed by the human. So in this one, because what is the plasmid? Plasmid are extra nuclear chromosomes, and these extra nuclear chromosomes are providing they are not vital, they are not required for vital mechanism, but they are providing extra characteristics to that bacterial cell in which they are present. Now in this one, if plasmid, we are using these plasmid as a vector. So if that plasmid providing extra characteristics, so what is that? That means they are providing bacterial cell to resistance to antibiotics. Now if they are providing resistance to antibiotics, then these antibiotics these sequences have a specific place then on which their restriction enzymes are working, and on that restriction we are able to utilize that restriction enzyme to cut that DNA and incorporate our gene. So in this one, that is the restriction enzyme, and that restriction enzyme is utilized to cut that plasmid, and that plasmid is cut by the at the tetracycline resistant place. Okay. Now, ninth one is most important cause of biodiversity loss. So they are very simple question. So that is the loss of habitat loss and fragmentation. Now, 10th question is which of the following is our example of mutualism. So what is that mutualism? Look at this one. There is a population interaction, and in that population interaction, because if those population which are living in same area, they are interacting with each other. When they are interacting each other, so in that interaction, if that interaction is very much useful for both the population, so that is known as mutualism. Now look at this answer. What is that answer? Which of the following is an example of mutualism? So lichen, mycorrhiza, plant and pollinator, both A and B. Okay, so that answer is both A and B. Now look at this one. This lichen is the combination of algae and fungi. So both are benefited. Mycorrhiza is a relation between fungi and higher plant roots. Okay, that is the higher plant roots. So that is the combination between two. Now look at this one. This one is a very much important one. Plant and pollinators. Pollinators means those which are helping in pollination. So plant, and if on the one side there is a animal pollinator, so if there is a option for animal pollinator, that means both are benefited. Now in the option of pollinators, wind is also working as a pollinating agent. Water is also working as a pollinating agent. So because of this reason, this option is not correct one. Now the 11th one, mammals from colder climates generally have shorter ears and limbs to maximize heat loss. The law is known as. So which one is the correct answer? Allen rule, founder effect, both A and B, and none of the above. So look at the answer, and that answer is none of the above. Why that none of the above? If you can convert this word into minimize, then the answer is first one. Then the answer is only one word is changing, and that is maximizing. So if you change the word minimize here, then the answer is that A. Otherwise, if you are not able to convert this this word and it remain maximize, then there is none of the above answer. Now next one, which of the following is an example of in situ conservation? So that is also from biodiversity, and in that in situ means conserving that species or that organisms in their particular habitat, and that is the answer is of sacred groves, because they are the artificially developed by the human being. Now that is the second half of the section A. And in the second half of the section A, there are assertion reasoning type of questions, and in that assertion reasoning type of question, you have to select the answer. So in this one, look at this one. Zytenogamy is functionally a cross pollination. So zytenogamy is part of plant embryology. Transfer of pollen grain, reason is transfer of pollen grain from the anther to stigma of another flower of the same plant. Another flower of the same plant. So it is a example of cross pollination. And what is the answer? Select from those one. Select from those parts. Look at this one. These are the options given, and out of these options you have to select one. Okay. So in this question, thus A is the correct one. Both A and B are true, and R is the correct explanation of B of A assertion. Why? Because the geitonogamy is the transfer of the pollen grain on the flower of the same plant, but that is the not the same location. That is located maybe of different branches or different site. Okay. Now next one is the 14th question. When two genes in a dihybrid cross means the cross between the transmission of two characters at a time. Okay. So when the two genes in a dihybrid cross are situated on the same chromosome, that means both the genes which are for different character, they are located on the same chromosome. The proportion of parental gene combination is much higher than non-parental, because linkage is working. Higher parental gene combination can be attributed to crossing over between two genes. Okay. So that is the more one, and A is the answer one. So next one is 15. Primary transcripts in eukaryotes are non-functional. Look at this one. There is central dogma, and in that central dogma, there are the process is making the copies of mRNA from DNA is known as transcription, and in that transcription, that mRNA is known as primary transcript. After that primary transcript, that is taking place, there is a taking place of processing, and in that processing their introns are removed. Okay. And that is converted into a working mRNA, and because look at the region, methyl guanosine triphosphate is attached to 5' end of hnRNA, heterogeneous RNA, because that mechanism of RNA processing is taking place. So right answer is C. A is true, but R is false. 16.1 is a community with more species is more stable than that with less species. So that community is the group of different population living in a particular area. So more number of species, the lesser variation in the total biomass production in the year after the year. So that is the both A and true, and R is the correct explanation of A. So with this one we are completing the section A. Okay, so section A is on the basis of multiple choice type or assertion reasoning type of questions. So we will go to the second B option, B section, and in that B section, look at this. Now the questions are of 2 marks. The graph given below show the variation in the levels of ovarian hormones during various phases of menstrual cycle. So look at this one. There are 2 hormones, and they are exhibiting the level of these 2 hormones at this menstrual cycle. So identify what is the question in this one. So look at this one. What is the question? That is the question. Identify A and B hormone. So look at this one. That is the A hormone. This one is the B hormone, and there is to the top of A hormone is reaching at the level top in the days of 14th one, and that B hormone reached to the top at the approximately at the 22nd days. So identify these 2 A and B hormones. So that is the question. Identify A and B hormone. So that is the hormone estrogen, and B hormone is progesterone. Second one question in this one is specify the source of hormone marked in the diagram. So estrogen is secreted by which organ, by which structure? That progesterone is secreted by which structure and which organ? So that is the A hormone. Estrogen is secreted by maturing ovarian follicle or Graafian follicle, and B hormone is secreted by the corpus luteum. So that is the first question of section B. Second question is both Down syndrome and Turner syndrome are example of chromosomal disorder. Cite the difference between 2 at the chromosomal level. Now, in eukaryotic cell, every cell has a fixed number of chromosomes, and we can say every species has a fixed number of chromosomes. Like human species or in human cell, there are 46 chromosomes. If that 46 chromosome number is changed, so what is that condition? So that Down syndrome is caused because of trisomy in the chromosome number 21. So in a normal cell, the cell is 2n, and when the cell is 2n, that means there are 46 chromosomes, and in that 46 chromosome number, 1 is present twice, 2 is present twice, 3 is present twice, so on. There are 23 chromosomes. Okay, so every chromosome is present in the number in 2. So if 21 number chromosome is present in number 3, one more extra chromosome, that is known as trisomy, and because of this trisomy, the Down syndrome disorder is created. Now, because of this reason, the chromosome number become 47. On the other side, the Turner syndrome is due to monosomy. That means in the women there are 2 X chromosomes, because out of these 23 chromosomes, the 23rd set in the female, that is XX chromosome, and in the male there is XY chromosome. So in the female, if one X chromosome is lost, so that condition is known as monosomy, and because of that monosomy, the disorder is known as Turner syndrome, and that the result of this Turner syndrome is that the 45 number of chromosomes, the one chromosome is decreased. Now, 19th question, your classmate complains of headache and cough. The doctor confirms that he is suffering from pneumonia and not common cold. So there are difference between the pneumonia and common cold. On the basis of certain symptoms, list these symptoms, mention any 2 precautions to following to prevent the spread of this disease. So you have to identify the different symptoms of pneumonia. On that those symptoms that doctor has declared the pneumonia that is person has suffered. So there are the features. Doctor confirms pneumonia on these features, and these are the features which are for common cold, and you have to take these 2 precautions to stop the spread of these diseases. Now, next question is, now that is from the biotechnology, and as we already discussed, that is the artificial plasmid developed by the human being. In PBR 322, foreign DNA has to be introduced in tetracycline region. Why? Because this one is plasmid, and that plasmid contain the tetracycline resistance gene from the restriction enzyme. Now, if that contain the tetracycline resistance gene and we are utilizing that as a vector, so we are able to incorporate that gene inside the inside this plasmid. So that we have to cut at a single particular place. So there is a restriction enzyme given below, which one should be used and why. So which type of restriction enzyme we have to use? So YPU first, echo R1 BMHI. So which one we have to use as a vector? That restriction enzyme. Second part of this question is, give reasons why these 2 cannot be used. Okay, so first one is that why you are utilizing that restriction enzyme. Second is, out of these 3, why you are not utilizing other 2. So look at this one, because why you are utilizing? You are utilizing this enzyme to cut the tetracycline resistance gene in this PBR 322 plasmid. Why? Because that restriction site is present. That restriction site of the that cut site of this DNA is present in the tetracycline region. So you are utilizing the BMHI restriction enzyme. So why not we are utilizing these one? Because there is no restriction site for these enzymes in the PBR 322. So because of that reason we are not utilizing this one. So that is the answer. Not used as a restriction site for this enzyme is present on other maybe on the other plasmid, and that is because of the ampicillin resistance, and EcoR1 that site is not present suitable marker for tetracycline resistance. So because of the presence of restriction site we are utilizing that particular restriction enzyme to cut that particular plasmid. Now next one is, that is from very beautiful one. So that is the from ecology. Compare the two ecological pyramids. So in that ecological pyramids are that hierarchy of organization of ecological hierarchy of the organism according to their eating habitat, and they are organized in the ecological pyramids of biomass given below, and explain the situation in which this is possible. So look at this one. There is trophic level and there is a dry weight. There are two ecological pyramids, because there are three type of ecological pyramid: pyramid of number, biomass, and ecology that is energy flow. So there are three type. Now look at this one. Also construct an ideal pyramid of energy if that is 200,000 joules of sunlight is available. Now look at this one, because these pyramids of number, these pyramids of biomass, these pyramids of energy, they are organized according to flow of energy, and this one is giving the amount of energy that you have this amount of energy, and make a diagram, make a ecological pyramid of these organisms according to this one. So that is the dry weight of these organisms. They are giving the dry weight of these organisms, and that is the different level of a ecological pyramid. So arrange these ecological pyramids according to this energy. Now what is that? How to write the answer? Now look at this one. There is a 10% energy rule. There is a 10% energy rule. If that whatever be the amount of energy is present in a ecological system, and that from sunlight, 10% of that energy is transmitted into the further trophic levels. Okay, so that 10% you have to apply that 10% trophic, that 10% energy flow rule to solve this one. So because if after applying this one is the answer. Okay, so first pyramid of biomass corresponding to a terrestrial ecosystem. Second refers to a small standing crop of phytoplankton supporting large standing crop of zooplankton or that aquatic ecosystem. So look at this one. This one is the pyramid, and that pyramid is of in the terrestrial ecosystem, because the primary producers are more. There are primary consumers, that is secondary consumers, and they are of top animals, that is a tertiary consumers. And on the other side, that is the aquatic ecosystem, and in that aquatic ecosystem these are the primary producers. They are less in number, and these primary that they are phytoplankton. On the other side, this one is the primary consumers, and they are the primary consumers. That means higher number of primary consumers are depending on less number of that phytoplanktons in the aquatic ecosystem. So arrange this one according to this, that that is the sunlight energy available. So that out of this one, 10% is converted in the form of energy or biomass in the primary that is producers. That out of these producer 10% is transmitted to primary consumers, 10% to the secondary consumers, or 10% to the top consumers. So that is the way you have to solve, and that is the way you have to write the answer. Now, 22nd question is differentiate between spermatogenesis and oogenesis. So, that is the part of reproductive system. So, spermatogenesis is taking place in the testis, and oogenesis is taking place in the ovary. So, that what is that difference? So, that is spermatogenesis. So, spermatogenesis is formation of a sperm, and how that spermatogenesis is taking place. So, that is the first difference. In spermatogenesis, continuous production of a sperm took place from puberty to old age. Now, because of hormonal effect, there are 13 years of age. After that puberty age, there is start the formation of that sperms in the testes, and that is because of hormonal effect. On the other side, that is oogenesis, that means formation of that egg. So, that is in the age, whereas in oogenesis the oocytes are generated before birth because they are formed through the germ cells, and these germ cells are found. These germ cells are found in the reproductive organs. These the oocytes, number of oocytes at the time of a birth of a female child, the number of oocytes already formed in their ovary, and in that ovary the number is fixed. After that one, that oocyte is converted into the egg during the puberty. So, that is the first and foremost difference. First, because spermatogenesis, germ cells are present in the testis, they are not converted into the sperm. They are converted into the sperm during the formation, during the stage of puberty. On the other side, all the oocytes are formed, that is the fixed number of cells are formed in the oogenesis before the birth of the fetus. Now, second one is large number of these oocytes are degenerated during phase from birth to puberty. Well, yes. Why? Because they are at the time till the development of puberty, or till the female child reaching to the stage of puberty, these are destroyed. So, the maximum numbers are destroyed. The oocytes continuously decrease in number, because at the time of birth of that female child, the number of oocytes is already developed. So, that whenever there is a destruction, so that number is keep on decreasing. It gets completely exhausted at menopause. Look at this one. In the male, the sperms are continuously formed, and these germ cells are continuously formed. So, if the sperms are continuously formed, so they are formed in that male at a different age, but at the time of birth a female child, the oocytes number is fixed. So, after a age, the oocytes number is completely reduced, and when that is reduced, that stage is known as menopause. So, in the female, that they are facing the problem. So, that stage is known as menopause. The third difference is primary spermatocyte complete the first meiotic division, leading to formation of two equal size secondary spermatocytes, whereas now look at this one. These are germ cells, and in these germ cells there is a division of meiosis. And because of that meiosis, the haploid cells, and they are known as gametes, are formed. So, that meiosis, now look at this meiosis, that meiosis is meiosis first. There are two division of meiosis, meiosis first and meiosis second. And because of this meiosis second, after meiosis first, there are formation of two cells. So, first cell is converted into two cells. In these cells, there is a meiosis second. So, in the meiosis there are 4 cell formation. So the first cell, when the first meiosis is taking place, there are 2 equal cells are formed during the formation of spermatogenesis or formation of sperm, while on the other side when the meiosis first took place in this one in the oocytes. So if this is the germ cell in the female oocytes, first meiosis is taking place, then the formation of 2 cells. So one cell is complete large cell and another cell is polar cell. So because of that reason there is unequal division during the formation of oocytes, whereas primary oocyte divide unequally by the first meiotic division, result in the formation of a large haploid secondary oocyte and a tiny polar bodies. So that is the division difference in the division, and that difference is because during the formation of gametes there is a meiosis, and in that meiosis the 2 division took place, meiosis first and meiosis second. And when the meiosis division first formed, there is a formation of 2 cells. So how these 2 cells are formed? If these 2 cells are formed during the formation of spermatocytes, while that is the unequal division during the formation of oocytes, okay, in the development of oocytes. So after this one the secondary oocytes are formed and one is the polar body is formed. So that is the 23rd question. Draw labeled enlarged view of microsporangia of an angiosperm. State the function of its innermost wall. So that microsporangia where microspore formation took place, and that is the answer. So look at this one. The outermost layer is epidermis. After that one there are 4 to 5 layer middle lamella. Middle layer is present, and after this one the innermost layer, that is the layer, and that innermost layer is known as tapetum. And after this one the microspore mother cells are present, and that microspore mother cells are divided by meiosis division and they form the microspore. So what is the second part of this question is, what is that tapetum is the innermost layer of the microsporangium. So what is the function of this tapetum? That tapetum provide the nutrition to the developing pollen grains. Okay, so this one is the question. Now that is the C section, and in that C section first question is 24th. Given below is a single DNA molecule. Frame and label its sense and antisense RNA molecule. Look at this one. It is not asking about the sense or antisense strand of DNA. So it is giving that is the sequence of a DNA strand, and it is asking about the sense or antisense strand of RNA, not the DNA, because what is the antisense strand in DNA, that is different, and what is the sense and antisense strand in RNA, that is different. Okay. So second part of this question is how the RNA molecule made from above DNA strand. That means you have to made a RNA from this strand and help in silencing of specific RNA molecule. So look at this one. So that is the sequence, and that is the sequence. So you have, that is a very simple one. There is no problem. You have to convert. This one is the sense strand, because look at this one. This one is the sense this one is the sense strand. So we have to find first that is the sense strand. So this one is the anti-sense strand of DNA. So this one is the anti-sense strand. Now if look at this one, during the transcription, because this antisense, that sense strand is expressing in the form of protein. So mRNA is containing the same sequence of this sense strand. And what is the difference? Instead of this thymine, there is a sequence, and that sequence is known as uracil. So that is the they are having the same sequence. You have to change this thymine into the uracil, and that become the sense strand of RNA molecule. Now change the sequence. Instead, now because in RNA instead of thymine uracil is present, so you have to change this. If you are making the antisense strand of this one, so that T is always attached with the A, so that A is always attached with the T. So in the T, the uracil is present instead of thymine, so you have to convert this one is the antisense strand of the RNA. So how you can convert this RNA molecule? Because RNA molecule is always present in the form of single strand, okay, and that maximally that RNA molecule, if it is present in a single molecule, it is working as a single strand. So if you convert it into a double strand, then you are able to convert this RNA as a inactive one. So two strand of RNA, that is the sense and antisense, being complementary, will bind with each other and form a double strand RNA. As a result, its translation and protein expression would be inhibited. Okay, so because of this reason, the RNA is working as a single stranded molecule, and if you are converting that RNA into double strand, then that create a problem, and that is not able to express in the form of translation or therefore the ultimate part is the protein expression. So one is here is a very beautiful diagram, and in that beautiful diagram, look at electrode, and there are some molecules. Water is here, heat source is there, two vacuum pump is also there, condensers are present, and all the water is present. That means there are two electrodes are present. So that is a very beautiful diagram, and in that diagram, look at what about that diagram. So in that diagram, different chemicals are present, and if different chemicals are present and these chemicals are heated up, so that is must be about something, and that is about the Miller experiment. And that Miller experiment, in this one there are three questions. State the hypothesis which S.L. Miller tried to prove in the laboratory with the help of setup given. So what is that setup and how that is what is that setup is explaining. Second part is name the organic compound observed by him in the liquid water after running the above experiment. Third one is a scientist simulated a similar setup and added methane and ammonia and water vapor at 800 degrees centigrade. Which important compound is missing in this experiment? A simple one. So you have to write this one. What is the Miller is proving by this experiment? So that is the chemical evolution, because from first form of life originated from pre-existing non-living organic molecule. First organic molecule are synthesized Big Bang, after Big Bang theory, and these organisms, because the life was originated in the water, and that is that water in the ocean is working initial stage working as a soup, and in that soup life was originated. So that is the chemical evolution. So that amino acids are synthesized, they are basic building blocks, and in this one the last one is hydrogen is absent in this one. Hydrogen is very much important. Now 26th number question. Humans have innate immunity, that means already present, have innate immunity for protection against pathogens that may enter the gut along with the food. What are the two barriers that protect the body from such pathogens? So that innate immunity means that immunity which is present already. So these are the two barriers which are protecting these these host organisms from the pathogens. Okay, so there are physical barriers, mucus coating of the epithelium lining the gastrointestinal tract help in the trapping microorganism entering into our body. So that is the part of innate immunity. And second one is physiological barriers, acids in the stomach, saliva in the mouth prevent the microorganism growth. So there are type of enzymes present in this one saliva, and that stomach is secreting HCl. So because of that is protecting that is protecting the host organism from the pathogens. 27th question is, biotechnology has helped the farmers to get pest resistant cotton crops. So that is the very much important plant that have developed by the human being by utilizing biotechnology, because plant is already present in the nature. Okay, so this plant is that is very much economically very much useful plant for the human being. So that is destroyed by the some insects. So to protect that plant from the insect, we have incorporated some gene in this one. Explain the technique adopted along with its mode of action. Okay, so what is that mode of action? We are utilizing biotechnology. In that biotechnology, we are incorporating a gene. That plant is damaged by the insects. So if we are utilizing that biotechnology, and in that biotechnology we have identified a gene, and that identified that gene very much playing a very much important role. So look at this one. The technique involves use of popularly known as biopesticides. Okay, and in that biopesticide Bt toxin produced by a bacteria, Bacillus thuringiensis. So that is bacteria. In that bacteria there is a gene which is producing a toxin known as Bt toxin, and that Bt toxin is affecting that insect which is damaging the cotton crops. So we are identifying a gene in bacteria. That gene is very much useful for the human being to incorporate in this crop. Second is, we have to separate this one. Now, how that Bt gene is playing the role? Bt toxin protein, when ingested by insect, that means this toxin is ingested by that insect, active form due to alkaline pH of the gut. Okay, that is ingested by that insect, and that reach to into the gut, and because of the alkaline pH. Now look at this. The activated after reaching to the gut, that is because of alkaline pH. It is converted into the active form, and that active form is able to bind with the midgut or midgut of the epithelial cells. Okay, after this one, look at the fourth point. It creates pores in the epithelial cells. This activated form of Bt toxin is binding with this one, and that creates pores in the epithelial cells. Okay, so that kills the insect, or that is very much important one. Why? Because we are requiring such a product which is destroying the insect but not destroying the plant, which is not affecting the plant or their any productivity. Okay, so that gene is very much useful. So we are separating this gene. Look at the gene which is known as cry genes, encoding this protein. Okay, so that protein is encoded by that cry gene. Protein are isolated from the bacterium. So that cell that is separated from the bacterium, incorporated into the crop plant like cotton. So cotton has a genome, and we are incorporating that cry gene into the genome of the crop plant. So the protein encoded by this cry gene control the pest. That gene, cry gene, after incorporation into the cotton plant, that is expressed in the form of protein, and when that is expressed in the form of protein, that means it is also producing that Bt toxin. When that is producing Bt toxin, that means that is also expressed in the cotton plant. So if the insect will eat that cotton plant or leaf part or any other part, that is creating same effect which is creating by the Bt toxin in the Bacillus thuringiensis. So that is the complete mechanism. And last point of this one is specifically cry A and cry that A second AB control cotton bollworm, and that is the pathogen which is an insect belonging to Lepidoptera, which earlier used to destroy the whole crop plant. So that insect is creating very much problem for us to destroy the cotton plant, and we are able to protect now that plant by developing Bt toxin variety of cotton. So that is the question. Now 28th question, assess the efforts of effects of loss of biodiversity in a region. So that is the simple question. So you have to mention any 4 effects of the loss of biodiversity. So you have to write this one: decline in plant production or decline in number of animal species, lowered resistance to environmental perturbations and such as drought, increased variability in certain ecosystem processes such as plant productivity or water use or pest or disease cycles. And 4th one is species may become endangered, increased rate of species extension. So they are the simple 4 effects of loss of biodiversity. Now next, that is from look at, very much beautiful, and that is about the hemophilia. Hemophilia is a sex-linked recessive disorder of humans. So that is hemophilia, and that is sex-linked recessive. So there are 2. That is sex-linked and that is the recessive disorder. 2 terms of human. The pedigree chart given below show the inheritance of hemophilia in one family. Study the pattern of inheritance and answer the following questions. So look at this one. There is a normal female, there is a normal male. They are marrying, and after that this marrying, this one is the generation. In this generation there are females are normal and there is a male. One male is affected and one male is normal. The female in this one is marrying to a normal person, and in that normal sequence, one is the offspring is one that male is affected and one male is normal. On the other side, there are 3 children. Out of that, all the women are normal and male is affected, and that normal women is marrying to a normal male. So that is the pedigree. So what is the question? So let us see what is the questions. Now look at this first one. So there are 1, 2, 4, 5, 6, 7, 8. They are from the same generation, and they are this 3rd one is from outside, 9th one is from outside. So they are marrying to the normal women. So go to this one. The first one is give all the possible genotype of the members 4, 5 and 6 in the pedigree chart. Okay, so go through this one. So we have to give genotype of this one. This one is the 4, 5 and 6. What is the genotype? So look at this one. This one is normal one. this one is affected, and this one is also the normal one. So go to the this one. This one is a sex linked recessive disorder. That is the sex linked and recessive. Recessive means that is expressed only in the form of homozygous condition, or if it is present in the form of heterozygous condition, then the only one copy is present. So because of this reason, it is expressed as a recessive order. So look at this one. The second part of this one, a blood test shows that individual 14 is a carrier of hemophilia. That means that is carrier, that is not diseased. The member number 15 has recently married the member number 14. What is the probability that their first child will be a hemophilic one? So with the help of Punnett square method. So look at this one. The member 50, the blood test always show that individual 14 is a carrier one. So this one is a carrier. So that means the 14 one is a female. Yes, look at this one. The 14 one is a female, and that is female, and that female is a carrier, and 15 is marrying to a normal person. So what is that ratio of their first child to be hemophilic? So these are 2:1. So let us discuss the answer. That is the first genotype of member 4. That is that member 4 is female. Look, go back to this one. Look at this one. This member is that this one is the female, and when this one is the female, that means this female is maybe this female is maybe normal, because this is the normal expression, and on the other side maybe this female is maybe carrier. So this one is the genotypic expression of the fourth one. Now what is the fifth one? Because fifth one is male, and if it is male and it is expressing as a hemophilic then the male having the combination of X and Y. So that because of this reason it is having only one X chromosome. So that is the genotypic expression of male, and this one is the sixth one. So X and Y, because this one is the male and this one is normal. So that is the expression of X and Y. So these are the expressions of 4th, 5th and 6th individuals in this one. So that is the answer of A. That is the answer of A. Look at this one. These are the answer of A, because the sixth one is individual is normal. So this one is X and Y. Now second one is what is that second question? In that second question, look at this one. A blood test shows that individual 14 is a carrier of hemophilic. That is because it is a sex linked recessive disorder. So the female is working as a working as a carrier. So female 14 is working as a carrier, and that female having the 2X chromosome, and that female is marrying to a person which is marrying to a person. The member number 15 has recently married to a member number 14. So that member 15 is normal one. Okay. So look at this one. That is male, this one is female, and this one is normal male, and in that there is a formation of gametes. So there are two type of eggs are formed. One is egg in which the hemophilic gene is present, and on the other there is formation of two sperms, two type of sperms, X and Y. So that is the Punnett square method, and in that Punnett square method, look at this one. This one is the female gametes, and these are the female gametes. So these female gametes are the egg, and one each X and XH. On the other side, these are the sperm. These are the sperm. So look at this egg is fused with this sperm. So this one is the combination. This is, egg is fused with this sperm. So look at this one. This sperm is fused with this egg. This egg is fused with this sperm. So when there's the 14 number is the carrier women is marrying to a normal person, at that time these are the egg combination, these are the sperm combination. So this is the female, this one is carrier, this one is the female, this one is the normal one, this one is the male, but this male is hemophilic and this male is normal. So what is the percentage of this one? The percentage is normal is 50% male, and that is 25 out of this total percentage, 25% male is hemophilic. So that ratio, and that is the Punnett checkerboard method to study this transmission of hemophilic condition after marrying that carrier women with the normal being. Okay, so next question is, the graph given below represent their three categories of organism responses. So that is from ecology, and in that one there are external levels, and that is the internal level because of how they are maintaining. So there are responses of the organism to external these environmental conditions. So they are giving three conditions in L, M and N to cope with the stressful condition. Identify the categories L and M. So what is that L? That condition, because of this external conditions are increasing, the internal environment is also increasing. So in the L condition there is a change in the internal environment of the organism. On the other side, in the M, whatever be the external condition, the internal environment of the organism is fixed. So that is very much simple one, no problem. Given below the example of some of the activities performed by animals, categorize these activities into the appropriate kind of the organism response L, M and N. Which type of the organism L, M and N are shown in the graph with region. In summers we sweat profusely. Second one is, sometimes desert lizards bask in the sun and sometimes they move into the shade. So how, according to these responses, how they are classified. So look at this one. L is conformers, because the internal environment of L is keep on changing. So second one is, B1 is regulators. So to regulate the body temperature, the A1 is they are able to maintain their homeostasis, internal homeostasis. So they are known as regulators, and if they are changing their internal environment according to outside environment, or we can say on the other side, they are not able to maintain their internal environment, so they are known as conformers, to keep their body temperature constant by behavioral responses for coping with the variation in environment. So these one. Next question is, trace the development of megaspore mother cell to the formation of mature embryo sac in flowering plant. So that is from the development biology. Look at this one. Trace the development of megaspore mother cell. How that megaspore mother cell is converted in the form of a embryo sac. So that is first, that megaspore mother cell. That megaspore mother cell first is converted in the form of a megaspore, and that megaspore is converted into a gametophyte. So that embryo sac is the female gametophyte. So look at the process of formation of megaspore from the megaspore mother cell is called the Microsporangium. So look at this one. In this one is the female flower. In that flower, this one is the ovary, that is stigma, style and ovary, and in that style, in that ovary there are ovules. So these are ovules. In that ovule there is a development of megaspore. So in that there is a cell, and that cell, that is known as megaspore mother cell. In the ovule there is a development of nucellus, and in that nucellus, from that nucellus there is a development of a cell, and that cell is known as megaspore mother cell. So further development took place in this way. That is the megaspore mother cell, and that megaspore mother cell divide, and form the 4 megaspores in the ovule. So this one is the ovule, and in that ovule there are formation of 4 megaspores. Out of these 4 megaspores, these 3 are become the non-functional one, and this megaspore, which is located towards the micropyle, of this ovule, that is functional one, and that convert that female gameto, converted into the female gametophyte. So this one is the megaspore, and that is the functional megaspore. So look at this one. In the ovule, in that ovule there is a nucellus. In that nucellus, germ cells are differentiated, and that is known as megaspore mother cell. That megaspore mother cell divide by division and form the 4 cells, and these 4 cells are known as megaspores, and these megaspores are located in the ovule. So these megaspores, out of these megaspores, 3 are become the non-functional, and 1 which is located towards the micropyle, that become the functional one. So that megaspore develop further in the form of female gametophyte. So that is the first stage. The process of formation of megaspore from the megaspore mother cell is called Microsporangium. So that is completed in the form of these stages. So we have already discussed the first cell. That first cell is megaspore mother cell. That megaspore mother cell at the micropylar region of the nucellus divide and form the megaspore. So that megaspore is functional. Now second one is that megaspore which is present towards the micropylar end, that is converted in the form that remains in the active form and convert in the female gametophyte, and that is the embryo sac. So how that is converted? Look at this one. So look at this. This one is the nucellus, that is the outermost boundary. In that outermost boundary, that is integument in the ovule. This one is the megaspore wall. That megaspore is enlarged, and this one is that is the nucleus of megaspore. That megaspore nucleus is formed after meiosis. So this one is haploid. So that megaspore nucleus, when germinate, first division is mitotic division, and in that mitotic division 2 nucleus are formed. So there after that one there is no cytokinesis. So look at this one. When megaspore divide, the megaspore nucleus is divided by the mitotic division, and in that one there are 2 nucleus are formed, and these 2 nucleus reach to the opposite poles. After this one there is another division. In this one there are 2 nucleus from mitotic division. So there are 2 nucleus on this side, 2 nucleus on this side. After this one there is further 1 more division, and 2 nucleus another 2 nucleus are formed in this region, another 2 nucleus are found in this region. So ultimately in the germinating megaspore or in the developing megaspore, which is converting into the female gametophyte, there are 8 nucleus, 4 nucleus on in this side, 4 nucleus on in this side. Out of these 4 nucleus, one nucleus is moved to this side, one nucleus is moved to this side. So out of these 4, one is here and one is here. So these one is lost. So there are 3 nuclei. Ultimately 3 nucleus remain on this side and 3 nucleus remains on the micropylar end side. So what happens to these nucleus? Look at this one. The nucleus of the functional megaspore divide by mitotic. So there is mitotic division, 2 nuclei which move to the opposite poles, forming the 2 nucleate embryo sac. So there are two nucleus in this one, and out of these 2 nucleus there are formation of mitotic division, and during that mitotic division there is a formation of 8 nucleate stage, and in that 8 nucleate stage 1 nucleus from each side reach to the center. Ultimately, after 8 nucleate stage, cell walls are laid down, leading to the organization of the typical female embryo sac, and in that typical embryo sac there are 8 nucleus and 7 cells. So what is that ultimate organization of that embryo sac? These 3 cells are grouped together, micropylar end, and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergid and 1 egg cell. The synergids have special cellular thickening that may be the filiform apparatus. So what is that ultimate organization of this embryo sac? So look at, we are making the final embryo sac of this one. This one is the megaspore, this one is the nucellar wall, and in that one this is the covering of the megaspore. So integument. There are 3 nucleus. These 3 nucleus are organized. One, the middle cell, that middle cell is known as egg cell on the micropylars, and there are on the this one is the egg cell, and there are 2 cells here. So these 2 cells are known as synergids. So there are 2 synergids. On the other side, on the opposite pole, that is chalazial end, and on the chalazial end there are 3 cells, and these 3 cells are known as antipodal cells, and in the center there are 8 cells. There are 2 cells, and these 2 cells are known as polar nuclei. Okay. So this one is the ultimate development of female gametophyte, and the female gametophyte or embryo sac, that is also known as embryo sac. Female gametophyte is also known as embryo sac. So that female gametophyte is made up of 8 nucleate conditions. So these are 3 nucleus, these are 3 nucleus, and these are 2. 8 nucleus and 3 celled condition, 7 celled condition. So 3 celled on this side, 3 celled on this side, and they are considered in 1 cell, the polar nuclei. So that is the development of female gametophyte or embryo sac from the megaspore mother cell, all the stages. So in that stage, from first the megaspore mother cell is converted in the form of megaspore, and that megaspore germinate or megaspore further development in the female gametophyte, and in the angiosperm that female gametophyte is known as embryo sac. After this one, the 32nd question, observe the representation of the genes, and that is the lac operon. So look at this one. This one is lac operon given below. So there are 4 options. Identify the region where the repressor protein will attach normally. So that is given by the operon concept, and in that operon concept that all the genes which are expressing in the form of protein, they cluster together, which are functioning in a similar way, or those genes which are utilized for similar function, they cluster together, and in that clustering, that is known as operon concept. And there are 4 options here. So identify the region where the repressor protein will attach normally. Second one is under certain condition repressor is unable to attach at this site, explain. So third one, if repressor fails to attach to the site said site, what product will be formed by Z, Y and A gene. Analyze why this kind of regulation is called negative regulation. So that is very much important one. So first one is operon. So whatever be the gene which is expressing in the form of protein, that means that gene contain the regulator sequences, that gene contain all those sequences which are helping that gene for the transcription. Now in this, these are the structural, these three are the structural genes and they are expressing in the form of enzyme. So these are structural gene, that is the promoter and that is the operator, that is the inhibitor gene and this is the P gene. So this I gene is forming that repressor protein. Now look at this one. This we are discussing about the lac operon. That means lac is utilizing, lactose is utilizing for the destruction. That means lactose is degraded in the cell. So whenever there is a presence of lactose in the cell, at that time this gene is expressed. So otherwise this operon is in the off condition. Why? Because there is no lactose, so there is no degradation of lactose. So to express these proteins, lactose must be present in this one. So first answer A, and that answer is operator region. So that repressor always attaches on the operator one. Second one is in presence of inducer. I am already saying that it is expressing only in the presence of lactose, because those genes which are expressing in the form of enzyme, they are expressing because they are requiring to degrade the lactose. If lactose is not present, then how they are expressed? So that is the answer, and that what the structural gene which are synthesizing three different type of enzyme, they are presenting, these are the different type of these three are the enzyme which are synthesized by the lactose genes, that is the Z gene beta galactosidase, Y gene permease and transacetylase, and that is the reason which is why it is called a negative regulation. Why? Because lac operon is expressed only when it is expressed lactose is present, and if lactose is absent, then it is not expressed. So this one is the reason. Because of that reason it is known as a regulator, that negative regulation. And in this section the last question is the 33rd question. Some restriction enzyme break a phosphodiester bond. Look at the restriction enzyme break a phosphodiester bond on both the DNA strand. That means we are talking about restriction enzyme, and these restriction enzymes are we are utilizing as a molecular seizure, and they are cutting the DNA. So in this one we are utilizing bomb H1, and that bomb H1 is utilized to incorporate a DNA. So what is the question? What is the objective of this action? क्या जरूरत है इसकी? And second one is explain how the genes of interest is introduced in the vector, not in the genome. So these are the two one. So the third one, you have given the DNA sequence, that is the DNA sequence one. If this DNA was cut with this restriction enzyme, how many DNA fragments would you expect? How many they are formed? Write the sequence of these double stranded DNA fragment with their respective polarity. Last one is a gene M was introduced in E. coli cloning vector, that is the vector at bomb H1 site. What will be impact on the recombinant plasmid? Give a possible way by which you could differentiate non-recombinant to recombinant plasmid. So that is very much important. That is only way to the antibiotic resistance gene. You have to change the antibiotic one. So first one is the answer. Two different DNA molecules will compatible to combine. Or second one is restriction enzyme cut the DNA of the vector and then ligate the gene interest into a DNA of the vector. So we have to convert that the DNA, cut the DNA and incorporate that DNA in the form of vector. So the last the third one is the how many fragments we are able to get. So there are 2 fragments. Why? Because that is bomb H1. So that is cutting the DNA from this side, and in that side, so because of that reason we are getting the 2 segments. So that is cutting the DNA at this side. Okay, so that is the one segment and that is the another segment. We are getting 2 fragments of the DNA. Last one is that bomb H1 site will affect tetracycline antibiotic. Now look at this one. How we have to identify this one? The plasmid is carrying the antibiotic resistance gene, and we are able to incorporate that outside gene or desired DNA sequence in the antibiotic resistance. So we can change the antibiotic resistance sequence of the DNA, and when we are changing the antibiotic that resistance sequence, at that time that will change the habit of that bacteria, okay, or habit of that cell. So we have to separate that plasmid, incorporate that gene into the antibiotic resistance and the sequence, and that is the tetracycline. So after incorporating that desired gene into that tetracycline resistance gene, the plasmid is not able to provide that facility to cell to become resistant. So if it is not able to show that resistance to that antibiotic, so we are able to identify that one. And in this one there are huge number of other methods are also done because of color change. That is the one example, and after this one there are many number the color change. Because in this one, if that cell is died, then what is the use of identification of that cell? So now they are utilizing color change. So they are asked about this one. So that is the answer, and because of this reason we are able to identify that organism or that cell from the culture media. Okay, so that is all about that model paper. So please write down the answer near about this one. Select the keywords from this one and select these concepts. Make a concept around these keywords. That is the most important one to write the subjective answer.